Hi, this is Trey Lath from OpenHelix. Today's tip of the week is on SNPxGE2. It's a database for human SNP co-expression associations. So let me read this for you here. The database contains computationally predicted human SNP co-expression associations, and as they explain, the differential co-expression between two genes is associated with the genotype of a SNP. This is actually quite a fascinating area of study. And this research came out recently in bioinformatics, as you see here, the paper, and it's also linked on the blog post. What they've done is they did a large-scale association study based on HapMap data, as you see here, covering 269 individuals from 4 populations, and did a computational analysis to find out if there was SNP and co-expression between two gene associations. Now, they put this data into this database here, and the data in the database is all from this paper in this study. So hopefully we'll find more of this in the future. There's 2 simple ways to search, and I'm going to go to the tutorial to show you. They have a printed tutorial. There's a quick search, as you see here. You can put in a gene symbol or a SNP ID. And then the step 2 here is then you click on an icon. We're going to do that real quick to get more detailed information. Advanced searches, you can take 2 genes and find out if there are any SNPs related to the co-expression of those genes. And it also brings you to the brief information page. Another example here is you can put in a gene ontology term and select a false discovery rate, which is related to p-values. And there's also a link in the blog there to tell you more about FDR. And here, the last one, you can put an exact gene or SNP ID to search. They also have a standalone program that you can download if you wish. I'm going to show you that link in a second. And there's more information in the help section here. So let's do a quick search. First, let me show you the links up here. There's Home, that's where we were just a minute. The Advanced Search, which we'll go to in a second. And the Download, I'll click that to show you where you can download the thing here in the tutorial. So let's go through this a little bit. So I could put in a gene of interest and see if there are any other genes that are co-expressed and that is affected by a SNP. There's associated SNP with that. So I'm not going to do that. What I'm going to do is I'm going to take you to the Advanced Search, and we're going to put in 2 genes. So I'm going to say clock and Pearl3. Let's say Pearl3. You see, I've done that before. And now you can give a more strenuous FDR cutoff, stringent, FDR cutoff here. Now, as it showed in the tutorial, you can also put a gene ontology term in, or ID, a term ID, and an exact search of a gene symbol. But we're just going to put in 2 genes here and submit. As you see, there are SNPs, or is a SNP, that is associated with the co-expression of clock and Pearl3. Now these 2 are circadian rhythm related genes. This is also found in the African population, so let's take a look at that a little bit closer. So once you click that details page, you will find more information, the score, the p-value, the FDR, the associated gene ontology terms, here signal transduction, transducer activity, and you'll find the co-expression data here and the information and links to take you to the information about those genes here, clock, Pearl, and the SNP. The function of this SNP is unknown. So this SNP is associated with the co-expression of clock and Per3. This will give you some more information about that SNP that you can now further do some greater discovery. So that's this very small but excellent database and data. Check out for today, and look forward to seeing you next week.